All right, welcome to installment 3 of Muscles. It's about time, and I want to start this out with a question. Have you ever tried to wonder about why it is after somebody drops dead, or a dog or a cat or a squirrel drops dead, they get stiff, I mean really stiff, and they go into the world of rigor mortis? Maybe you haven't, and that's okay. But maybe you've wondered, watching some type of a forensics TV show, how is it that somebody can tell how long ago somebody died? And there are a lot of different ways to do it, but one of the things you do is measure what state of rigor mortis they're in. So this rather morbid entry into this video brings in a few new players. Calcium and sodium, they're going to play an important role in this whole process of muscle contraction, because that's what we want to look at. We've laid out the anatomy essentially, but now we have to play around with the physiology. Calcium, sodium, that should be an Na. Calcium and sodium are going to be important players in this. Both calcium and sodium, these little ions that we're going to be dealing with, have a positive charge. Charge is important. I tried to really emphasize that early in the semester. You'll see why today, and more so when we get to the nervous system. We're also going to deal with a big molecule called acetylcholine. It's a neurotransmitter. It's how neurons will talk to the muscles. Now, before I erase this and I bring up the projector, I want to point out this. I'm going to be talking about things called action potentials. I'm going to try and gloss over them a bit, because we'll talk about them in detail about 2 videos down the line when we get to the nervous system. So, yeah, action potentials are essentially, it's a spark, it's electrical discharge, it's a nerve impulse. And it can travel down a neuron, it can travel across membranes or down membranes. So you might end up with a lot of questions about what was he talking about with these action potentials. It's okay, we'll cover them in detail, I think 2 videos down the line. So that being said, let's get back to becoming stiff after you die. Let me see if I can get this turned on again. There. This is essentially where we left off. We had the cell membrane that you know, called the sarcolemma, fancy name for just a phospholipid bilayer. And then we have these extensions that go into these things called T-tubules. And then we have this big network, the sarcoplasmic reticulum, a modification of the endoplasmic reticulum that you are also familiar with. And then we've got these big packages of myosin and actin, and this is where the business is going to happen. Now, when I left off, I said that, hey, calcium plays a big role. Calcium is what's going to be important when it comes to trying to get the actin and the myosin to bind, to do what we call a power stroke and move them across each other, move them next to each other. So we've done this. Now let's look at the physiology, and I'll try and make it about as quick as I can. This is what we want to do. We want to take these sarcomeres and we want to bring these Z bands or Z lines closer together from both sides. So we've got our myosin and actin, myosin, actin, myosin, actin that are interacting together. We've got the troponin, the tropomyosin that's keeping this from happening. So let's take a look at the neurons themselves. These are what we call end bulbs off of what we also refer to as motor neurons. Motor neurons are neurons that are going to be innervating with your skeletal muscles, well, your muscles, and they're going to be essentially telling the muscle to contract. This is where the signal is going to come from. So anytime you write notes, you blink your eyes, you do this on the board, you are taking in impulses from the motor neurons into the muscles themselves. Here's what they look like in an artist's rendition. You can see the spinal cord, which is the central nervous system. Here we've got the peripheral nervous system innervating with all of these myofibers. In some cases, one might just innervate 5 or 6, maybe with the eyes, where you do really fine movement. But maybe if it happens to be the quadriceps or the biceps, you could have 400 or 500 of these myofibers. And you're looking at massive movement with one impulse. But I'll try and address that again a little bit more when I'm not so rushed in the next video. Here's what this actually looks like when you get right down to the end. Here's that end bulb. So here's our motor neuron coming down. And if you look, there's this gap, this cleft. They don't actually touch. There's this little thing called a synapse that's in between. So the only way this can talk to this, the neuron can talk to the myofiber, the muscle, is by taking the contents of these little vesicles and exocytosing them. So spew out the contents of them. What will be in these is acetylcholine or acetylcholine. That will exocytose and it will cause this cell down here to respond. I'll wait to get back to this. So essentially this is what happens. The action potential, this mysterious spark that comes flying down here, this nerve impulse, is going to get to the end bulb. Calcium with a positive charge is going to move in. You're going to have little tiny gates or channels that are going to be popped open, and the calcium will move across. Now here's something else that's kind of weird. All membranes have what's called a membrane potential. The charge on the inside is different than that on the outside, and we can measure it, and I'll show you when we get to the nervous system. Same thing here. This is different than this. This is different than this. And it's all in charge. It's all about ions, positive and negatively charged ions. When the calcium moves in, that's the stimulus that says exocytose the acetylcholine. The acetylcholine is dumped across and ends up now binding with the membrane of what we call the end plate or the muscle fiber. Now here's what's going to happen, and this is, I'll go through this hopefully not too quickly. What's going to happen next is once the ACH moves across and sends in the signal that says, hey, it's time to do something, sodium channels are going to open. So when sodium channels open, I'll do it up here because there's a little bit more room. Sodium will move into the sarcolemma. So it goes outside, now it moves in. And what happens is we get a change in the membrane potential. We get a change in the electrical charge. And it's what we call depolarizing. So we'll talk a lot about depolarizing, like I said before, when it comes to the nervous system. This is going to cause more gates, I'm trying to take this as slowly as I can, it's going to cause more gates to open to let more sodium in. So now the sodium that's coming in isn't because of a chemical gate, one that was opened up because there was acetylcholine there. But now these gates are popping open because the charge is changing, and they're what we call electrical gated. Here's the spark. Here's the action potential. And it continues. Let me keep this outside. Okay, put that in the T-tubule so it's still on the outside. So suddenly the sodium starts flowing in like crazy. And that spark, or that action potential, dives way down into the middle of the muscle fiber. So it's not just something that's happening on the outside. These T-tubules that I mentioned before bring that impulse all the way to the inside. Now, once they do that, remember, we haven't talked at all about our sarcomeres yet, but we're about to. This action potential shooting through, ripping through the T-tubules, this one, and the next one's off to the left-hand side, and these off to the right-hand side, they're going to stimulate the sarcoplasmic reticulum to release calcium. Now we can kind of get back to where we're comfortable. Once calcium floods out across here, remember this is a cut section, those little cisternae of the sarcoplasmic reticulum go all the way across. So suddenly they just flood everything in calcium. Well, you know the story. When calcium gets there, calcium is going to bind to TNC. TNC moves the tropomyosin. Once the tropomyosin moves, myosin can bind to actin. Our thin and thick filaments can finally do that right there. So we get this big stimulus coming in, this message that's gone from electrical to chemical to electrical, and finally the calcium is released. Now let's go to this. Let's start out up here. And what you need to do, this is a myosin head, and there's the tail. Take a look at the position of these, because not a whole lot changes in these images as we go around, but it's trying to keep a focus on the myosin. We'll start out up here. Now, we get the myosin and the actin have bound. So obviously the tropomyosin has moved away, but you really can't see that clearly, and they make the connection. Now look at this. It bends. It does this big shift in shape. That's what's called a power stroke. And when it power strokes, what it's going to be doing is sliding past the thin filament. Once we've done that, ATP, this energy molecule, we'll talk about more, ATP is going to bind and it's going to break the bond. And it's going to reset the position of the myosin. It's kind of normal to think, oh, hey, ATP is involved, that's energy. Well, that must cause the power stroke. It doesn't cause the power stroke. It breaks it, which is kind of weird. This will get us back to rigor mortis in hopefully about 3 minutes. Once we've done that, we've reset, we've taken the ATP, we burn it to get our ADP and a phosphate, still disconnected. Then they can connect again. The power stroke. New ATP comes in, we break the bond, it resets. And remember, there are millions and millions and millions of these little myosin-actin interfaces that are doing this power stroke continuously when you tell a muscle to contract. Now, what the cell wants to do is it wants to pull the calcium back into the sarcoplasmic reticulum. So it's trying to all the time. But if the impulse is coming from the neuron and that whole action potential thing is taking place, it kind of overwhelms it. But ultimately, when the impulse from the neuron, the action potential is no longer there, the calcium goes right back into the sarcoplasmic reticulum, that resets, and there's a molecule that's constantly at work called acetylcholine esterase, that's an A, that's constantly attempting to break down the acetylcholine in that little gap between the neuron and the muscle. So once you quit telling that neuron to kick out acetylcholine, kick it out, kick it out, kick it out, it's wiped out. And calcium then is immediately pulled back into the sarcoplasmic reticulum. That's how you go through a muscle contraction. Now, to wrap us up, let me get back to rigor mortis. Why is it when you drop dead, you stiffen up for a while? Well, here's what happens. You don't have ATP. You're no longer making it. Your cells no longer function. And if you don't have ATP, you can't break that bridge. So whatever state of contraction, whatever position the muscles were in, with no ATP, you can't relax them. So after a while, not too long, I'm not a forensic pathologist, I think it's a few hours, these start to degrade because they're not maintained, there's no homeostasis, and then you loosen up. But that's the story behind rigor mortis. So sorry, I went just over 15 minutes. I will be back with some extra odds and ends, like I said, twitches, possibly tetanus, and some other things that have to do with muscles. And I'll get that fourth one out there as well, ASAP.